So good afternoon, everyone. It's so nice to see so many of you here. I know I am personally very excited about today's lecture for a number of reasons. I guess I'm embarrassed to admit this. I've never heard our speaker speak before, so I'm really looking forward to Tom's lecture. But I think the topic at hand about discovering genes and the use of animal models, particularly zebrafish, will be very fascinating. So I'm very much looking forward to that. Before we get into that, let me just do a few housekeeping items here. First, I want to start off by thanking the wonderful folks in the Dean's office for the terrific job that they do making these events so successful and spectacular. So let's give them a warm round of applause. Second item is for those of you who have not heard, Money Magazine recently rated us as the #1 college in America, and we bumped Princeton off of that position. So I think that's really exciting news. So we should be happy about that and very proud. I'm not exactly sure what that means. I don't know if that necessarily means that we're better than Harvard. I think it does. So, these events are just so remarkable, and we're so appreciative to our ambassadors for helping support the school. So if you are interested in learning more about, providing some kind of support to the School of Biological Sciences, you might want to consider joining BioSci Ambassadors. There's multiple opportunities, multiple levels in which you can join. And if this is not your thing, please consider, liking us on Facebook, Instagram, Twitter, LinkedIn, and whatever other social media you are on, because that really helps us in spreading the word about what we are all about. So now let's get to the topic at hand, which is today's lecture. And so Tom Schilling is the chair and a professor of developmental and cell biology. He is a Pew Scholar and has also a fellow of AAAS. He holds appointments not only here at UCI, but at University College and the Imperial Cancer Research Fund, both of them of which are in London, England. And as I mentioned earlier, so much of what we learned about gene function has come from studying simple organisms like zebrafish and other animal models. So let's give Tom a warm round of applause.
Thank you. So I want to thank Frank, and I'd also like to thank the whole staff of the Dean's office for putting this on. I really like to thank everybody for coming, and I realized a couple of weeks ago that this was probably going to come up against the 7th game of the World Series. And of course that has come to pass. And so if you're not at the reception and you're a baseball fan, I don't blame you. I also like to thank my department staff, and everyone who's helped me along the way. And what I'm going to tell you about today is some of the work we've done discovering genes that control development and disease using my favorite system, the zebrafish. And before that, I also want to thank all the current members of my lab shown here from a recent road trip we took to a meeting up on the Central Coast, as well as all my former lab members shown here, as well as many undergrads who are in the lab. I think that's too much. So I want to start with what I want to call one of the grand challenges of biology, and that is really how do we bridge different levels of complexity with all the advances that have been made in sequencing genomes, new techniques for visualizing proteins and networks within cells, for actually watching cells as they assemble into organs and organisms. How do we understand how things at one level work together to achieve the next level? And on top of that, there's the environment influencing at every level. And so it really is a grand challenge. And what we do know, a lot of what we do know has come from work in model systems like yeast, worms, nematode worms, flies, mice, And each of these has its own distinct advantages in terms of, for example, having a relatively small genome, relatively few cells, where you can really understand all the components going into the assembly of a given organ, or an organism that's closer to humans, where the organs are more similar to ours and where they get diseases that are more similar to ours. And so there is a need for systems that have that combine many of these advantages. And that's where fish come in. So fish, we evolved from lobe-finned fish that were very similar, presumably, to this living fossil, the coelacanth that you might have heard of. And the lobe-finned fish diverged from the common ancestor for zebrafish about 400 million years ago. But despite that ancient diversion, we still share approximately 85% of our genes with, or 80% of our genes with fish. And that's really one of the revelations of the genome era, of all the genomes that have been sequenced, that it's really not the new genes that make us human, but how those genes are deployed and regulated. So what I'm going to talk about is first how zebrafish has become a very popular model for human development and disease, particularly for gene discovery and for imaging. It's really a great system for imaging. And then touch on some work from my lab looking at the musculoskeletal system, which is our main interest, how you build a face, particularly a skull, and birth defects associated with that, and then also a more recent project studying tendon development and the role of mechanical force, of the environment on that process. So usually when I tell someone that I work on zebrafish, I get one of two questions. One of them is, can you eat them? And I usually say no, but then looking online, I find that people actually do, or they pretend to. Even these transgenic glowfish that are fluorescent in sushi, I don't think I would eat that. Or why zebrafish as opposed to some other fish or some other vertebrate organism? Why are zebrafish so useful? So I'm going to get a little bit into the weeds of some of the details here that you might not understand all the details of. But these are some of the real advantages of zebrafish. So like some of those simple systems like worms or flies, you can house a lot of zebrafish in a small space. They breed year-round, they lay lots of eggs, and they're cheap to maintain. And you can go from function to gene. Because you can house so many animals, you can do things like genetic screens where you randomly mutagenize genes and look for defects in the fish and then clone the gene down the road. Or, as you may have heard of, you can knock out specific genes with things like CRISPRs, or even knock in fluorescent tags into genes and make transgenic fish where you can track cells in the living animals. They're vertebrates, so they have a well-annotated genome sequence where it's quite easy to identify human relatives of genes in the fish and do comparative genomics. They have transparent embryos, as I'll show you, and that means you can track cells and molecules in the living organism with fluorescent proteins. And you can also do something called optogenetics, which is a relatively new technique where you can make transgenics that allow you to look at live cell activity in the living organism. There's a whole society now of zebrafish disease models because they get birth defects, have cancers, and many neurological disorders that are very similar to humans. And one unique advantage of this system, because they live in water, is you can do drug screens, a relatively high throughput, especially on the embryos and larvae, by adding small molecules to the water. So here to illustrate the first point, see if I can play this, are some breeding zebrafish in a lab in New Zealand. And they're a couple inches long. If a new student comes to the lab, we usually teach them how to tell a female from a male. There's a male swimming by. Put them in a tank over a rough surface, and they lay eggs, and those are these little 1 millimeter clear circles here. And the rough surface is good because they fall through, and then the fish can't eat them, which they tend to like to do. And in the next segment, you'll see one of these eggs developing into an embryo. The cells divide on top of a big uncleaved yolk. They then wrap around the yolk. And within 24 hours, you start to see things like the eye and the muscle segments and the tail extending off of the yolk. And then it starts twitching around inside of its chorion, its shell. And then by the end of this movie, it's about ready to hatch. And because it's transparent, this allows all kinds of amazing images, especially at these early stages. And this is a transgenic fish where all the nuclei of the cells, I really think if I turn this off, I still hear ringing, yeah? Can I? I can't? Okay. This was actually a movie that was in a science paper from about 10 years ago that really blew us all away. So in this is using a light sheet microscope, which we now have one on the UCI campus, to visualize the embryo developing starting at the 64 cell stage, where all the nuclei of the cells are labeled. And you can track all the cell divisions as the cells divide on top of the yolk. And because it's using a light sheet, we can see it in 3D, the whole embryo. So this is two views of the same embryo. One where the head is going to come towards you over the yolk, and the other where the head's going away from you. And so you can see cells dividing and converging into the body axis. Here's the tip of the nose up here, and slowly you start to see things emerging like the two eyes here on either side of the developing nervous system, or the muscle segments will start to appear over here. So for example, if you were interested in the eye, you could take a movie like this now and track every cell back to its, through its ancestry to the beginning. Let's see, I've got another movie from this. And so this then allows you, let's go to the next one. To really quantify cell behaviors in unparalleled detail. So you can do things, especially at these early stages, like track the movements of all the cells as they first spread on the yolk and then converge into the body axis. You can follow cell division patterns, so you can find asynchronies as they emerge in this field of cells. You can look at the orientations of the divisions in every part. Okay. And start to see how these asymmetries emerge in different parts of the animal, as quantified here in this paper. Good. And then the last thing, the fish made a big splash last year, no pun intended, by being the first animal to have every one of its cells, at least the embryo, interrogated for its gene expression using a technique called single-cell RNA sequencing. And then using computational methods, that information was reassembled into the lineages of all the different cell types, at least in the embryo. And this science ended up highlighting this as the 2018 breakthrough of the year, and it surpassed all the breakthroughs in physics and chemistry. So we were really quite proud of this as a field. Okay, so one of the interests in my lab is how you build a face. And fish have a face that only a mother could love, especially if you shrink them down for scanning electron microscopy. But they have a face that has most of the components that ours do. And clearly there's a strong genetic component to the face, as evident by the similarities between relatives in a family. And there's also potentially genetic component to why people look like their pets. right? Maybe there's some genes that lead to these, two getting together. Not sure. But we all recognize faces very readily, and a lot is not known about how genes control the assembly of the skull or the face. And when I first took a job at UCI, I came with a collection of mutants in fish that had been randomly generated by chemical mutagenesis in a lab in Germany. And we were interested in cloning the genes underlying these different fish with funny faces. So these are larval zebrafish. Here's the eye. Here's the jaw. These are about 5 days post-fertilization. And some of these mutants have essentially no jaws. Others have sort of flattened heads or pushed-in faces. And my lab and others over the years have cloned the genes underlying these mutants, and all of them turn out to be good models for human birth defects and syndromes. Things like branchio-oculofacial syndrome, auriculocondylar syndrome, which affects the ears and the skeleton, mandibular hypoplasia, small jaws, or more general defects of skeletal biology. And so one of those that I'm going to highlight today, it was really a novel pathway at the time, which is a signaling pathway using a small protein called endothelin-1, and mutations in endothelin-1 or its receptor or something called phospholipase C all turn out to cause a birth defect called auriculocondylar syndrome. And I'll come back to that. Okay, and studying this in a fish really has the advantage that fish retain the simple segmental organization of the head. So fish have gills, and those gills are segmental homologs of the jaw and the jaw support. And fish retain these large bones, here shown in red and blue, of the upper and lower jaws, dorsal and ventral one of the first segment, or the jaw support, that in our skeleton, we have a lower jaw, and then the homolog of this dorsal bone is one of our middle ear bones. And when we stain an early fish larva for cartilage or bone and look at it like this, you can see how simple the early skeleton is. So if you roll a fish over that's stained for cartilage here, so this is the lower jaw labeled 1 here, and these are the eyes, you can see that each cartilage, which will later become a bone, is a simple linear row of chondrocytes. And so with such a simple system, you can really ask questions about how cells contribute to one segment and not another, how they contribute to one bone or another, in a lot of detail. And these structures all form in the embryo from a transient population of cells that you may have never heard of called the neural crest. And these cells migrate more than just about any other cell type in vertebrate embryo from their origins near the brain, near the hindbrain here, different segments of the brain, down into the face. Here a schematic of an early vertebrate embryo, and this will be the eye here. They're going to form all these skeletal structures of the face, including all these bones shown here, as well as the middle ear bones. So what we can do in a zebrafish is make a transgenic where green fluorescent protein is expressed in all of these neural crest cells in the embryo, and then make a movie and follow them. So here's a movie made by Pravir Sharma, a postdoc in my lab, this is a nuclear targeted green fluorescent protein, where this is the forming eye here, and you could just see, I'll do that one more time. You can see these cells that started out over here migrating and spreading over the whole head of the animal. And so you can take a movie like that. Pravir has done this with the neural crest, and then you can track the cells. So let's see if I can get this one to go. Where you either do this manually or have a computer track individual nuclei, and then having the nuclei labeled helps because you can easily distinguish one from the other, and you can start to build maps of how they change neighbor relationships, how their origin relates to their final destination, and really do it for every cell. So by being able to track the cells, we can start to see how different domains that will give rise to different bones emerge. And so for the jaw, what we found is that these early domains that will give rise to the upper jaw, called dorsal here in yellow, or the lower jaw, which is ventral in red, or an intermediate domain that gives rise to the joint between them, is already specified, and the cells are already kind of in place by about a day and a half after fertilization. And those structures will give rise to the upper jaw, the maxilla, or the mandible, the lower jaw. And so then getting back to this secreted molecule endothelin, what we found was that while, I lost a panel, but a couple of panels. So anyway, the endothelin, which was known for a long time as a small 21 amino acid peptide that was involved in vasoconstriction and inflammation and a bunch of other pathological conditions, also acts as a patterning molecule that determines upper and lower jaw identity. And from further work, we really showed, have strong evidence for endothelin actually acting as a gradient with higher levels inducing lower jaw identity and lower levels inducing upper jaw identity. And furthermore, looking at our mutants, we realized that the loss of endothelin led to something called a homeotic transformation within the jaw. And this is a concept called homeosis that was coined by William Bateson, who was a famous English scientist at Cambridge, who also coined the term genetics for studies of inheritance. And Bateson realized that it was not uncommon in biology to see transformations of one structure into another. So for example, it's well known that homeotic selector genes can transform a segment of a fly that would normally form a small balancer organ called a haltere into a segment that makes a full-blown wing. So you can get a four-winged fly, whereas a normal fly only has two. Or in plants, you get homeotic transformations between petals, sepals, and stamens, all different directions. And most of the plants that you know of that have huge numbers of petals, the really elaborate flowers, are homeotic transformations. So in the case of the jaw, what we found was that here's the pattern in wild type. With too little or no endothelin, you lose the ventral structures of the jaw. But if you increase endothelin signaling, you can actually get a homeotic transformation of the upper jaw and the dorsal part of the second segment into duplication of the lower jaw. Seems like kind of a crazy result, right? But based on the fish studies, human geneticists who were mapping birth defects not that long ago mapped a rare birth defect called auriculocondylar syndrome to phospholipase C, which had been shown in fish to cause this homeotic transformation. And these kids have a small jaw, usually a tracheotomy, and misshapen external earlobes. And they said in the paper, by reading the fish papers, they realized when they looked at the CT scans of these kids that they actually have a homeotic transformation of the lower jaw into an upper jaw-like structure. And then later, in another rare birth defect in humans, mandibulofacial dysostosis with alopecia, it was realized that in fact there's a homeotic transformation in the other direction. So they have their upper jaw, their maxilla, looks more like a mandible. And this has not only implications for these two rare birth defects, but also for how the defects in many other birth defects that are more common, like cleft palate, may reflect at least partial homeotic changes in identities of cells. And one of the things we found in the zebrafish that's kind of exciting for potentially doing something about a problem like this is that because endothelin is a small peptide that's secreted extracellularly, we could take human endothelin protein, which is commercially available, and inject it into the face of a fish. And if you get the dose right, you can restore the jaw back to normal morphology. So that gives it some promise as a potential therapeutic for something as structural as a jaw defect. And finally, and this is more for the aficionados, but because we can track every cell and we know a lot about the gene regulatory network surrounding endothelin signaling in these different domains of the face, we can build models, and we are building models that consist of all of the cells in this structure in a zebrafish, as well as many of the gene interactions that we know about. And our initial models, together with Ching Ni's lab in the math department, have been able to recapitulate some of the things we see experimentally, like the loss of the ventral structures in endothelin mutants and the rescue of the normal pattern when we restore endothelin. So then I just want to, before I switch gears, I just want to tell some of the researchers in the room some of the other things we're doing in my lab, including looking at other graded signals, like retinoic acid and how it sets up different domains within the brain. We're coming back to that breakthrough of the year. We're doing single-cell RNA-seq of neural crest cells to look more at their heterogeneity and how different lineages emerge in this population. We're looking at cell polarity within cartilages and how this impacts how cells stack into these linear rows and how that affects shaping of different bones. And finally, we actually are using a couple of different species of fish, some African cichlid fish, in collaboration with Tony Long's lab in ecology and evolution, to try and map genes that control facial shape in fish, using hybrids between two species of fish that have completely different facial shapes. Okay, so that's kind of complicated. We're going to come back now to a little bit simpler notion. So in addition to studying genes that control these things, we're also interested in the effects of the environment. And we've gotten recently interested in the roles of mechanical force in musculoskeletal development and disease. And all cells in your body are experiencing and have experienced force, including force that's exerted upon them from the extracellular matrix and the space around them, as well as junctions between cells, particularly in endothelia that are tightly holding on to each other, or even loose cells like blood cells experience shear stress as they move through blood vessels. And one of the extreme examples of stress in a system on cells is with your tendons and ligaments, which are constantly being exposed to force from muscle contraction, and obviously it's really important for movement. And tendons attach muscles to bones. Ligaments attach bones to other bones. And defects in muscles and tendons are devastating, and it turns out very poorly studied in most respects compared to tendons, at least, compared to muscles or bones. So inherited disorders like muscular dystrophy can lead to muscle atrophy, but also tendon injuries, tendon and ligament injuries essentially will affect all of us at some point. I've already had a few of them. And really there's very little or relatively crude treatments for these because we know so little about the cell biology of tendon cells or the regulation of the tendon matrix. So a postdoc in my lab who's here, Arul Subramanian, had worked on tendons in flies for his PhD. And so when he wanted to come to the lab and see if he could start studying this in a zebrafish. And he cloned a gene that is related to a gene important in flies for tendons called thrombospondin 4, and made an antibody to it, and it labels in red here all the muscle attachments in the fish, shown in the head. Here's the eye, and these are the in green are the muscle fibers. And with this antibody, really showed that the thrombospondin 4 localizes to muscle attachments here in just one small part of the fish, in these segments where the body wall muscles attach to the segment boundaries. And interestingly, this protein becomes concentrated at these segment boundaries as the muscle fibers elongate and start exerting force on those attachment sites. He then knocked out thrombospondin 4. So in these animals, there's no protein. And initially, it didn't look like it did much because their muscles seemed to be attached just fine. But then once he exerted additional contractile force on those muscles, they started to detach and snap like rubber bands away from the attachment sites. And then really showed that it was due to thrombospondin 4 specifically by injecting thrombospondin back into these fish, and you could rescue those attachments. And furthermore, here again, you can buy commercially available thrombospondin 4, human thrombospondin 4. If you inject that in the protein into a thrombospondin mutant fish, you can also rescue these detachments. And furthermore, if you inject more of that protein or inject that protein into a normal fish, you can make its tendons stronger. So they're more resistant to additional contractile force. And a collaborator, really, who's interested in rare forms of muscular dystrophy that he thinks are probably more tendinopathies, tendon defects, has shown that thrombospondin 4 also localizes around muscle fibers in human tissue and is one of the most highly expressed proteins in human tendons. So the last little bit of science I want to show is we're interested, so thrombospondin 4 is a secreted protein in the matrix of tendons. We're interested in the cells that make that matrix, the tendon cells or the tenocytes. And so recently, in collaboration with a lab at Harvard, we've been looking at a transgenic fish. This is a live fish in which all of its tenocytes are expressing a red fluorescent protein. And here's a fish that's stained for thrombospondin 4. You can see that the patterns are very similar, and in green are the muscles. So now we have a fish where we can really track these cells and study their development in both with imaging in live animals, and also we can isolate the cells and evaluate all kinds of things. So the one last experiment here that Arul did was now take these fish and image them and paralyze them, either using a mutant that's paralyzed from birth or injecting them with a drug that blocks neuromuscular transmission so that they're temporarily paralyzed. And then when he imaged these tendon cells, these tenocytes, he found that they form these tree-like branch structures that no one had ever seen before that extend these long processes into the matrix. In a normal animal, they're about 100 microns long. If you paralyze the fish, they shrink. This is color-coded. So now they go from red to blue is about 100 microns. Here, yellow to blue is sort of like 2/3 that. They shrink by about 1/3. And then if you restore force by stimulating muscle contraction, that restores the length of these processes. So we think that these are actually tension sensors, force sensors, that are monitoring the force and potentially modulating the matrix and allowing a tendon to adapt to the force that it's exposed to. And one thing we're hoping to do in the future to get at this is to collaborate with a lab at UC Santa Barbara that has made a tension sensor that involves implanting small beads, droplets of oil, and then imaging the deformation of the oil droplet as a way of measuring the tension that the surrounding cells are putting on the oil droplet. And he can also put magnetic particles in these droplets and use a magnet to exert force on surrounding cells. So one slide that I like to show is from a friend, a colleague at University of Washington who tore his ACL in a soccer game and got pictures from the keyhole surgery of the before and after. It looks pretty bad before, and the fix is to take a tendon out of his hamstring and sew it in to his knee. And this is the current surgical treatment for something like this, and it works, but it really ignores all of the subtle differences between the type of cells and matrix that go into a anterior cruciate ligament and a hamstring tendon. And I think with further knowledge of really differences between cells in different structures like this and how they adapt to force and regulate matrix production, we can come up with much better therapies for these kinds of injuries. So then just to come back to the grand challenge, what I've told you is that we can use a system like zebrafish to gain insights into gene functions, things like endothelin-1, as a small molecule that can pattern the parts of the skeleton, or thrombospondin, which is important in tendons. Because of the simplicity of the animal, the embryo, we can really bridge the complexity of cells to organ in the sense that we can watch every cell and quantify how it contributes to something like a bone or a tendon. And we can also use the system to get at the impact of certain things in the environment, like mechanical forces. So that's it. And I want to thank everybody in my lab that's done all this work, collaborators at other institutions, collaborators at UCI, at least the ones I've talked about, and there are many others, and I really want to say how much I've benefited from the community and environment at UCI and many other collaborators that have helped me along the way. Particularly a lot of collaborative things that I've done on the computational side with Ching Ni and our growing zebrafish group at UCI. So we've got 2 other fish labs here now. And since I started as a graduate student, the community has expanded enormously so that there are really labs at just about every major research institution. So thank you.
Very nice talk. I have to say I'm very grateful you listed me under humans. Most other people would have put their deans under the worm category. So we have time for some questions, and we have some microphones here, so if you raise your hand, Narita or Lizzie will be sure to track you down. But I guess while we're waiting, let me start off with the first question, and that is, can you elaborate a little bit more about the scientific breakthrough of the year, and what did we learn about doing all the RNA-seq in the zebrafish embryo?
Well, by being able to sequence all the RNAs in individual cells. So you can dissociate all the cells of the embryo, and then the real breakthrough was the ability to evaluate all the RNAs in one cell. So now you can say, all right, if I compare this cell to this cell, how many differences in its gene expression can I recognize? And then using computational methods, you can determine which cells seem to have a younger gene expression signature versus an older, and then build them into these trajectories where you can see all the intermediates as they work their way towards that lineage.
Okay, Peter?
Tom, thank you for that lovely lecture. In the first story you talked about where you said the human geneticists had looked at your work and seen parallels with the development of the human jaw, you also had some information about they also had a phenotype in humans which was overgrowth or overexpression of that pathway. And I wondered, I have 2 questions about that. One is, can you recapitulate that phenotype in fish by manipulating the genes of the fish?
Yeah.
And the second thing is, could you maybe talk about the power of the fish as a genetic system to really then kind of get at other genes that impact that pathway that could then inform the human geneticists about how that structure develops in humans?
So yeah, the simple parallel to that overexpression has been published by Chuck Kimmel's lab. So if you inject more of the protein, you can lead to the flipping such that the upper jaw becomes a lower jaw. And the working out of other components, so several other components of proteins that interact with the endothelin receptor. We did some studies of the receptors, and things downstream of the receptor were either discovered in fish or in mice, and then placed into the endothelin pathway through epistasis experiments. And so those become new candidates for other birth defects. And there are other birth defects out there that are superficially similar to auriculocondylar syndrome, but haven't been diagnosed as such.
Do those genes map to areas that in humans might be associated with that phenotype?
Not that I know of yet, but it's only sort of 4 or 5 years that people have been looking. The question was whether those genes were related to other birth defects in humans with similar phenotypes. Enrico?
Yeah, really nice and very interesting presentation. Can you give us an idea of how important are post-translational modification, lipids, many things that are not in the RNA or genetic information as a starting, but all of the factors that you need in order to grow an organism?
Well, that's a big question. I think an example of that is that the endothelin starts out as a much larger transcript and translated molecule that then gets processed post-translationally down to this little peptide. And some of the proteins, enzymes that are required for that processing, when mutated, also give craniofacial defects. And some of those we found in our fish screens. But globally, in terms of how important, I think it's just as important as transcription. And, that's another level of complexity that needs to be bridged.
Diane has a question.
Hi, Tom.
Hi.
So you said that the ability to apply drugs in the aqueous solution is a way to screen for new molecules that might be effective in treating particular diseases. Are there specific categories of diseases or drugs that have been particularly benefited from the use of zebrafish, and are any of them associated with these types of developmental defects?
So none of us, I've thought about doing drug screens to look for things that affect skeletal development, but I haven't done it. But there are some amazing examples. The first one that comes to mind is Lenzon's lab at Harvard. Harvard Medical School has been doing this for cancers, and both using like FDA-approved libraries of small molecules, making transgenic fish for particular oncogenes and other cancer-causing things where larval fish get the cancers. And then he puts these libraries of small molecules on those fish, which are small enough you can put them into plates, 96 well plates, and look for the cancers in the individual, either the onset of the cancers or the spread of the cancers. And he's done this for various types of leukemia in particular, but he's also gotten interested in melanoma, so fish get melanomas, and he's found some drugs that work on, that reduce the incident or spread of melanomas. And he's also he did a drug screen for things that enhance that was the first slide I showed actually. My intro slide was one from one of his Nature papers where he showed, they found a drug that enhanced the engraftment ability, essentially a bone marrow transplant experiment in a fish, and showed that drug enhanced engraftment of hematopoietic progenitors, and that's now in clinical trials in humans. Yeah.
So, Tom, can you elaborate on what kind of neurological disorders are being modeled in the zebrafish?
Well, there was someone here recently who was working on epilepsy. So they get seizures, and he was doing drug screens and had a whole bunch of hits with drugs that would reduce the seizures in the fish. There are people working on neurodegenerative disorders. I don't know much about those studies, but yeah.
Okay, any other questions? Oh, yeah, Peter.
Tom, in the news recently, over the last year or so, there's been a lot of talk about genetic engineering and CRISPR-Cas9 as a method to modify the genome. And I wondered if you might tell us about how that has affected your ability to make mutations in fish or put reporters into fish, and how that's kind of sped up maybe the pace of research for you in the fish community.
It's really been revolutionary. We've really suffered through multiple rounds of ways of doing targeted. We don't have embryonic stem cells in zebrafish, so we can't do the types of selection that you do in a mouse and then make a mouse from that. So we've always relied on trying to target them with things like zinc finger nucleases, and then these talons, and in both cases, those were quite tricky for the average lab to design these molecules such that they would knock out a targeted gene. So CRISPR-Cas9 is something that is so much easier in terms of the molecular biology, and there are these sites online that will help you find the right sites to target in the genome, and there are just many more targets than there were for these previous methods. And so it's really CRISPR-Cas9 has made it possible for small labs without a lot of expertise in the technology to make their own targeted knockouts and transgenics.
Could I ask a follow-up to that? And that is, one of the things that the people who work with zinc finger nucleases say is that they have less off-target effects.
Right. So now we're into that era of many of the CRISPR-induced mutations that people have made, where they had knocked down the gene's expression in the past and gotten, the phenotype they were looking for. When they use the CRISPR, they don't get it. And so the question is there compensation when using CRISPRs? And the zebrafish community is really struggling, having lots of workshops at meetings and things, trying to compare notes on what kinds of compensation people are seeing, what's underlying that compensation, and how can we get around it.
And I guess the other question that follows from that, which is relevant to maybe in the long run people thinking about using CRISPR-Cas9 genetic engineering tools to engineer the human genome, is from the experience in zebrafish, what can you say about the off-target effects of CRISPR? So when you go to knock out a gene, could you mutate something else, which of course if you did it in a human could have drastic effects?
Not much yet. I don't think we have enough data from the labs that are interested in trying to quantify that I could say anything meaningful, really.
All right, let's take two more questions on the right-hand side and then on the left.
It's just a general question. So humans, have a placenta, and they go through a great deal to try to protect development from infection. And I'm curious what happens if you take lipopolysaccharide, or LPS, and inject that into a zebrafish while it's beginning to divide. Does it have cells that are sensitive to that, like a human would?
I'm not sure I can answer that question. I think I've seen a talk about that, but I don't know the answer. Yeah, sorry.
Lee?
Yeah.
It's a two-part question. Part one, who are you rooting for the World Series?
Washington. Yeah.
Okay.
Underdogs. Yeah.
Part two, when it comes to birth defects like craniofacial defects in humans, what percentage of those do you think is genetic versus environmental? I know that's a hard question to answer. But.
Well, the big one, the one that affects like 1 in 400 live births is cleft palate, cleft lip and palate. And that one is still a real tough nut to crack. So it's clearly most forms are polygenic. And you know, twin studies and things like that suggest that many of them have a very strong genetic component, but what the genes are is tough. You know, there have been a few. IRF6 was one that underlies a large number of clefts. So cleft lip and palate are two different things. Sometimes they go together, sometimes they don't. And IRF6 was a big breakthrough from human genetics about 10 years ago, and now people are realizing, kind of like the endothelin pathway, that going from IRF6 upstream and downstream, they're finding other good candidates to underlie cleft palate. There are a lot of other really rare ones, you know, like auriculocondylar is more like 1 in 50,000, something like that. And those are clearly very much genetic. So, but cleft palate is the big one at meetings and stuff that people are really going after now.
Okay, one more question.
In one of the videos you showed the cells migrating from the neural crest into the face and into the head. And I was wondering, not knowing much about that, if you did this repeatedly with different embryos, how consistent is that pathway that each of those cells take? Are you able to pinpoint a particular cell, let's call that cell Jack, and follow Jack from one embryo to another one? Does Jack follow the same pathway each time? Are there statistical correlations?
Right. So, yes, some cells, for example, people have shown that there are, in certain parts of the neural crest, there are leader cells and follower cells, and the leader cells are unique. If you delete them, that causes a problem for the rest of the stream of migrating cells. Probably a lot of the followers are interchangeable and undergo slightly different migratory trajectories. As populations, they're very stereotyped. So those streams that come down from the sides of the head are exactly the same in every embryo. And we did publish one study of development of the palate where we showed that the two lateral parts of the palate come from a stream of cells that go under the eye, and all the cells in the middle of the palate that are missing in cleft palate actually go over the eye, and they kind of sew themselves together later on. So there are various very stereotyped aspects to migration, but it's not necessarily going to be the case that the same cell does exactly the same thing every time.
Okay, so I just want to make a couple quick points. Our next distinguished lecture is going to be given by Craig Stark, and it will be focused on why our memories decline with age, and Craig is here. Craig, you want to raise your hand? So if you're nice to him, he might give you a sneak preview of what he's going to talk about. That'll be in the winter quarter. And please, make sure you sign up for our emails so you're aware of the dates and all the other wonderful activities we have. And before we break for our reception, let's thank Tom for a really fascinating and very interesting lecture.
Thanks for coming.
Thank you.